If you feel afraid, nervous, or simply don't know how to attempt graph-based questions in the joint CSIR-UGC DBT-JRF NET examination, then this course is for you. Many aspirants skip graph-based questions because they think they are difficult or time-consuming. But the truth is, graph interpretation is not about memorizing, it's about understanding a simple sequence of steps. In this complete teaching series, I will teach you graph interpretation from absolute basics. Even if you have never understood the terms like axis, variable, scale, slope, or intercept before, don't worry. We will learn every concept step by step using simple explanations and life sciences examples. By the end of this series, you will be able to read graphs confidently, avoid common mistakes, and solve graph-based questions much faster in the exam. So welcome to DeepInsight Biosciences, and let's begin our journey into the graph interpretation for life sciences and biotechnology. Topics to be covered in graph interpretation are foundations. We will start from the absolute basics of graph interpretation. You will learn the fundamental concepts needed before reading any graph. Then we will go to the roadmap, in which we will learn how to read a graph in a sequenced manner. Then comes the chart types. You will learn the different types of graph commonly used in life sciences and biotechnology. We will also discuss when and why each graph is used. In slope and intercept, understand what slope and intercept represent mathematically and biologically. These concepts help you interpret trends and rates correctly. In mathematical curves, learn how to recognize important mathematical curves and understand their biological significance. This will make complex graph much easier to interpret. In do not confuse concepts, we will compare commonly confused graphs, concepts, and learn how to distinguish them easily. This will help you avoid frequent exam mistakes. Coming to summary chart of similar looking graphs, we will create a quick comparison of graphs that appear similar but represent different concepts. This serves as a rapid revision tool before the exam. In comparison snapshot, we will learn how to compare multiple graphs quickly and identify their key differences. This skill is essentially useful for analytical questions. In biological applications, we will see how graph interpretation is applied in real life sciences and biotechnology topics. We will connect every graph with practical biological examples. In exam preparation, we will learn a systematic approach to solving graph-based questions in competitive exams. We will focus on accuracy, speed, and smart thinking. In worked examples, we will solve previous and model questions step by step together. This will show you exactly how to approach graph-based problems. In common mistakes, we will understand the mistakes students frequently make while reading graphs. Learning this will help you avoid losing easy marks. In results checklist, use a simple checklist to verify your interpretation before selecting the answer. This reduces errors and improves confidence. Coming to practice tips, learn effectively strategies to improve your graph interpretation skills through regular practice. Small improvements here can make a big difference in exams. In answers to focus, we will discuss how to identify the most likely correct answer by interpreting graphs efficiently. This is especially helpful for multiple choice questions. And the last one, sample questions, in which we will solve actually graph-based questions from recent CSIR-UGC NET examinations. This will help you apply everything you have learned in real exam setting. Now, before we learn how to read different types of graphs, let us first answer a very basic question. What exactly is a graph? A graph is simply a visual representation of the relationship between two or more variables. Instead of reading a large table full of numbers, a graph helps us understand the same information quickly and easily. On the left side, you can see two axes. The horizontal line is called the x-axis, and it is usually represents the independent variable, which is the variable that we change or control during an experiment. The vertical line is called the y-axis, and it usually represents the dependent variable, which is the variable that we measure as a response. Every point on the graph represents one observation or a piece of data collected during the experiment. When these points are connected, they reveal a pattern or trend. Now, a graph gives us three important things. First, it shows the data, which are the values collected during an experiment. Second, it shows the relationship, meaning how the dependent variable, that is y, changes with the independent variable, that is x. Finally, it helps us understand the scientific meaning or conclusion behind the pattern. One important point to remember is a picture is not automatically a graph. A proper graph must have clearly defined variables, labeled axes, and an appropriate scale. In life sciences and biotechnology, graphs are used almost everywhere, from enzyme kinetics, cell growth studies, drug dose response, population biology, ecology, molecular biology, biostatics, and so on. So, understanding graphs is an essential skill for every competitive exam aspirant. Now that we know what a graph is, let's understand the coordinate plane because every graph is built on it. If you understand this slide clearly, reading graphs becomes much easier. The point where x and y axis meet is called the origin and its coordinates are 0, 0. Every point on a graph is measured from this origin. Coordinates are always written as an ordered pair, which means x comes first and y comes second. So if a point is written as 3, 5, it means move 3 units along the x axis first, then 5 units upon the y axis. The coordinate plane is divided into 4 quadrants. Quadrant 1 has positive x and positive y. Quadrant 2 has negative x and positive y. Quadrant 3 has both x and y negative. Quadrant 4 has positive x and negative y. To remember the directions, moving right means x is increasing, moving left means x is decreasing. Similarly, moving up means y is increasing, while moving down means y is decreasing. In most biological graphs, the x represents the independent variable such as time, dose, or concentration. The y-axis usually represents the dependent variable such as growth, enzyme activity, or response. One of the biggest mistakes students make in graph interpretation is ignoring the scale. Before interpreting any graph, always check the scale because the same looking distance can represent completely different numerical changes. Let's start with the linear scale. Here, equal distances always represent equal addition. For example, if the x-axis is marked as 0, 10, 20, 30, 40, every step increases by 10. This is the most common scale used for parameters like cell count, height, weight, time, temperature, absorbance. Next is the logarithmic scale. Here, equal distances do not represent equal addition. Instead, they represent equal multiplication. In this example, each step increases 10 times from 1 to 10, then 100, 1000, and so on. Log scales are commonly used for bacterial growth, gene expression, viral load, antibody titer, pH, enzyme kinetics, because this value can change over several orders of magnitude. Now look at the broken axis. Notice the small break in the axis. This means a portion of the scale has been omitted to save space or highlight small differences. While this makes graph easier to display, it can also exaggerate visual differences if you don't notice the break. So before comparing slopes, fold changes, or values, make it a habit to check the axis labels and the scale first. Remember, the graph may look same, but the meaning can be completely different depending on the scale. From this point onward, we will follow one fixed sequence to read every graph. If you master these 6 simple steps, you will be able to approach almost any graph systematically without confusion. First, decode. Understand the title, axis, variables, units, scale. Then second, recognize, in which you have to identify the graph type from the chart family, curve shape, slope, and the intercepts. Third, quantify. Carefully read the values, compare groups, variations, median, mean, error bars, standard deviation, and confidence intervals. In the fourth, you have to interpret, where you have to convert the visual pattern into biological meaning and identify trends, relationships, and patterns. Coming to the fifth point, you have to evaluate, where you have to check whether the graph has any limitations or misleading features and assess statistical significance. And finally, predict, where you have to estimate the future values within the observed trend and predict only within the limit of the available data. In the next few slides, we will learn each of these 6 steps in detail, one step at a time. Now let's begin with the first step of graph interpretation, decode. Before you try to understand the graph or answer any question, first understand what the graph contains. Start with the title. The title tells you what the graph is about. Here the title is effect of temperature on enzyme activity. So we immediately know that this graph is studying how temperature influences enzyme activity. Next, identify both axes. The x-axis on the horizontal axis represents the independent variable, which is temperature. This is the variable that is changed during the experiment. The y-axis or vertical axis represents the dependent variable, which is the relative enzyme activity. This is the response that is measured. Now check the units. Temperature is measured in degrees Celsius, while enzyme activity is measured in percentage. Without knowing the units, you cannot interpret the values correctly. Finally, look at the scale. On the x-axis, each division represents 10 degrees Celsius, and on y-axis, each division represents 20% enzyme activity. Reading the scale correctly prevents calculation mistakes and wrong conclusions. So always remember this rule. Before reading the graph, first read the title, identify both the axes, check the variables, understand units, and verify the scale. Once you have decoded the graph, you are ready for the next step. Now let's move to the second step, recognize. After decoding the graph, do not read the values immediately. First, recognize the overall visual structure of the graph. The first thing to identify is the chart family. This is a line graph where data points are connected to show how a variable changes continuously. Next, observe the curve shape. The graph has a unimodal shape, meaning it rises to a single peak and then falls. Now look at the slope. From 0 to 40 degrees Celsius, the slope is positive because enzyme activity is increasing. Around 40 degrees Celsius, the slope becomes 0 because the graph reaches its highest point. From 40 to 60 degrees Celsius, the slope becomes negative as enzyme activity decreases. Finally, identify the intercepts. The graph has a y-intercept of approximately 3%, meaning enzyme activity is about 3% when the temperature is 0 degrees Celsius. In this graph, there is no x-intercept within the displayed range. Always remember, recognize the graph's overall structure first and only then move on to reading the next values. Now comes one of the most important steps in graph interpretation, that is quantify. The word quantify simply means to measure using numbers. Instead of saying treatment A looks better than treatment B, we now answer questions like by how much is it better? What is the average value? How much do the readings vary? Can we trust these results? Scientists do not make conclusions by looking at graph alone. They support every statement with numbers. Let's understand each part one by one. Look carefully at these dots. Each dot represents one observation or one experiment. Imagine 3 students writing a test 5 times. Control scored 40, 45, 50, 55 and 60. Treatment A scored 60, 65, 70, 75 and 80. Treatment B scored 32, 37, 42, 47 and 52. Instead of looking only at one number, we observe all the readings. After this, we have to measure the mean. The mean is what most people call the average. Imagine 5 children have chocolates. One child has 40, another has 45, another has 50, then 55 and the last child has 60 chocolates. Now suppose we collect all the chocolates together and distribute them equally. Every child finally gets 50 chocolates. That equal share is called the mean. For our control group, 40 plus 45 plus 50 plus 55 plus 60 equals to 250. Now divide by the number of observations. 250 divided by 5 equals 50. So the mean is 50. Treatment A mean is 70, treatment B mean is 42. On the graph, the yellow diamond represents this average. Then we have to look at the median. Median is nothing but the middle value after arranging the numbers from the smallest to largest. Imagine 5 children standing according to height. Shortest, slightly taller, middle, taller, tallest. The child standing exactly in the middle is the median. For control, 40, 45, 50, 55, 60, the middle number is 50. Treatment A median is 70, treatment B median is 42. Unlike the mean, the median is not affected much by one unusual large or small value. What is the difference between mean and median? Let us understand this by an example. Suppose 5 children receive pocket money. First child got 20 rupees, second child got 20 rupees, third child got 20 rupees, fourth child got 20 rupees, and the fifth child got 500. Then the average becomes very high because of the 500. But the middle value is still 20. So the mean gets affected by extreme values. The median doesn't. This is why scientists always compare both. If we are clear about mean and median, let's move on to the variance. Now look at the dots. They are not all in one place. Some are closer together, some are farthest apart. This spread is called variation. Variance tells us how much the observation differ from one another. Imagine 5 children throwing a ball into the basket. If all 5 balls lands almost at the same place, then there is low variation. If they land all over the playground, then there is high variance. The more spread of the data, the larger the variance. Let's move on to the standard deviation. Instead of saying the values are spread a lot, we use one number called the standard deviation. Imagine you throw darts. If every dart lands close to the center, the standard deviation is small. If darts are scattered everywhere, the standard deviation becomes large. On this graph, the sign and error bars represent one standard deviation. Sign and means it's one of the color. Longer bars means more variation, shorter bars means more consistent data. If we look at the difference between variance versus standard deviation, students often get confused here. Variance and standard deviation describe the same idea spread. Variance is measured in square units, standard deviation is measured in the original units. That makes standard deviation much easier to understand. That is why most biological graphs show standard deviation rather than the variance. We will learn about these statistical topics even more in detail when we will start learning Unit 14 methods in biology, in which you will come across this topic in statistics of that unit. Now at the last, confidence interval. Now look at the white error bars. This represents the 95% confidence interval or 95% CI. Imagine you ask 100 students about their average height. You cannot measure every student in India. Instead, you take a sample. The confidence interval tells us, based on this sample, the true average is most likely somewhere inside the range. Think of fishing. If you throw a net, you do not know exactly where the fish is, but you know it is probably somewhere inside the net. The confidence interval is like that net. A narrow confidence interval means we are more confident. A wide confidence interval means we are less certain. Now compare the groups. If we compare the averages, treatment A has the highest average response, control is in the middle, and treatment B has the lowest response. But do not stop there. Also compare the spread. Are the dots tightly packed? Are they widely scattered? Look at the error bars. Large error bars means more uncertainty. Small error bars mean more reliable measurements. So whenever you see a biological graph, don't simply look at which line is higher. Always ask 5 questions. What is the mean? What is the median? How much variation is present? How much large is the standard deviation? How confident are these measurements? Once you answer these questions, you have successfully quantified the graph. Now let's move to the fourth step, interpret. Up to this point, we have identified the graph, recognized the structure, and measured the values. Now it's time to answer the most important question, what does the graph actually mean biologically? First, observe the visual pattern. The graph shows a slow rise followed by a rapid increase, then a plateau, and finally a decline. Now connect each pattern with its biological meaning. The lag phase represents bacterial cells adapting to the new environment. During the log phase, cells divide rapidly, causing exponential growth. In the stationary phase, nutrients becomes limited and growth slows because cell division and cell death occurs at similar rates. Finally, in the death phase, the number of viable cells decreases as cell death exceeds cell division. The overall trend tells us that the bacterial population does not increase at constant rate. It changes continuously with time. The relationship shown here is between time and the log of viable cell number. Always remember, don't stop at describing the graph. Your goal is to explain the biological process behind the pattern. That is true graph interpretation. Now let's move to the fifth step, evaluate. Even if a graph looks convincing, don't accept it immediately. First, evaluate whether the graph presents the data fairly and whether the conclusion is actually supported. Look at this graph carefully. At first glance, it appears that the treatment has caused a huge reduction in cell viability. But if you look closely, you will notice that the y-axis starts at 90% instead of 0%. This is called a truncated axis, and it exaggerates the visual differences. Now read the actual values. The control group is 96%, while the treatment group is 94%. The real difference is only 2 percentage points, much smaller than the graph appears to show. Next, ask yourself whether enough information is provided. We don't know the sample size, we don't see the replicates, and there are no error bars to show variation. Without this information, we cannot judge how reliable the results are. Finally, remember that statistical significance cannot be determined just by looking at the graph. We need information about variability and appropriate statistical tests. So remember, a graph may look dramatic, but that doesn't necessarily mean the scientific conclusion is strong. Now we come to the final step, predict. Once you have understood the graph completely, you can make predictions, but only within the limits of the available data. Look at this graph. The observed data are available only up to day 8. If we want to estimate the biomass on day 5, which lies between two measure points, we can safely use interpolation. Here the estimated biomass is approximately 50 mg. Now what about predicting beyond day 8? This is called extrapolation, and it should always be done with caution. Beyond the observed range, the biological pattern may change, so your predictions become less reliable. Always remember, the predictions are strongest within the observed data range. The farther you move beyond the available data, the greater the uncertainty. So the final rule is simple. Prediction is an estimate, not proof. Never make conclusions beyond what the data can support. Before we end this foundation section, I want to give you one simple checklist. Whenever you see a graph in the exam, ask yourself this same 10 questions in the same order. If you answer them correctly, you will rarely misinterpret a graph. First, what does the title tell you? Second, what is shown on the x-axis? Third, what is measured on the y-axis? Fourth, what are the units? Fifth, what type of scale is being used? Sixth, what is the overall shape of the graph? Seventh, what are the important values? Eighth, how do the groups or trends compare? Ninth, what do the error bars or variation indicate? Finally, what can you conclude? And just as importantly, what cannot be concluded from the graph? Make these 10 questions your habit. Every graph will become much easier to understand. With this, we have successfully completed part 1, the foundations of graph interpretation. In this section, we have learned the basic concepts that every student must understand before attempting any graph based question. Remember, a strong foundation makes every advanced topic easier to understand. In part 2, we will move to the next stage and explore different graph families. We will learn how to identify each graph type, understand where it is used in life sciences and biotechnology, and how to interpret it correctly in competitive examinations. Thank you for watching. If you found this session helpful, continue with part 2, where we will begin our journey into the fascinating world of graph families. See you in the next video.